Hi, my name is Melanie Föll. I'm a postdoc in the lab of Olga Witek at Northeastern University in Boston. I will present mass spectrometry imaging in the age of reproducible medical science as a poster talk which complements my talk from the CompMS session. The work I present here has been done in the lab of Oliver Schilling at the Institute for Surgical Pathology in Freiburg. Mass spectrometry imaging, short MSI, measures the spatial distribution of thousands of analytes directly in a sample. MSI is used in many different applications, including preclinical and clinical research, such as drug and drug metabolite imaging, investigation of tumor heterogeneity, and biomarker discovery, to only name a few. A complex and time-consuming step in an MSI project is data analysis. One hurdle is the lack of accessible software that allows reproducible data analysis. Therefore, we integrated functionalities from existing open-source software such as Cardinal, Multiquant, and Scikit-Image into the Galaxy framework. As a result, 18 MSI-specific Galaxy tools are now publicly available via the European Galaxy server, the Galaxy toolshed, and a Docker container. Currently, we apply those MSI Galaxy tools to analyze a variety of MSI peptide imaging data from various cancer tissues. I would like to take the opportunity of this talk to highlight the MSI analysis of a bladder cancer tissue cohort in Galaxy. The cohort consists of 2 tissue microarrays, short TMAs, with formalin-fixed paraffin-embedded urothelial tissue cores from over 40 patients. The tissue cores stem from 3 different types of urothelial carcinoma tissues and benign bladder tissues. The study aims to discriminate tumor and stroma histologies as well as tumor areas that stem from infiltrating and non-infiltrating cancers. Peptide imaging was performed on a multi-TOF mass spectrometer with 150 micron spatial resolution. The complete analysis has been done on the European Galaxy server. Raw data was imported into Galaxy, spectra were annotated as tumor and stroma, and both files were combined. Quality controls and preprocessing were performed to remove technical noises and shifts. Afterwards, classification was performed and the most discriminative mass features were visualized. This slide shows the Galaxy workflow that performs teachmark-based co-registration and annotation of MSI spectra. This workflow enables the transfer of annotations from the optical image to the MSI image, which later allows for supervised statistical analysis of the MSI data. Before statistical analysis, technical artifacts were removed with the pre-processing workflow that was tightly regulated by quality controls between each step. Pre-processing was successful in decreasing intensity batch effect between the 2 runs, as well as in correcting for the mass error. We used the spatial Schrankencentroid algorithm from the Cardinal software for supervised classification. We classified tumor versus stroma histology as well as infiltrating tumor versus non-infiltrating tumors. We obtained high accuracy for both training and prediction in both classifications. Statistical regularization was performed as part of the classification and allowed to extract the most informative features. These were 69 mass features for the tumor versus stroma classification and 84 features for the infiltration classification. We took the 2 most informative mass features for each classification category and plotted the spatial distribution based on the normalized raw intensities in a few tissue cores. As expected, those features show different abundances in the different groups. The complete analysis has been performed on the European Galaxy server. We will publish our study together with the raw data and the used Galaxy workflows and histories to set new levels of transparency and reproducibility in the field of translational MSI research. I would like to thank all colleagues from the Schilling Lab and from the Witek Lab, as well as all collaborators from the Institute for Surgical Pathology in Freiburg, from the Bioquant Institute in Heidelberg, and the Galaxy Team Freiburg.